Okay, so I will start soon. Before starting, yeah, if you want to play, so we have some fun during this lecture, you can connect to paulev.com, ValérieV593. And I see that we have already 2 sleepy persons, 1 tired one, so that's a good start for the lecture. 1 is okay, 2 good, okay. All right, no sick, no mad, that's cool. And then, okay, so I hope I will not make you more sleepy. And I count on you to interact, ask questions, comment. Yeah, be dynamic, so it will keep you awake, and it will keep me awake as well. So, yeah, we have 2, 3, 4 minutes more. So some of you were too sleepy, I guess, to come last Thursday at 8 a.m., which I fully understand. So anyway, the first lecture was quite global. It was an overview of what we're going to discuss in the next one. So it was more about the general principle of why is it important to know something about cell specialization in a multicellular organism, what it is, cell specialization, and today we will go, and we also saw how transcription factor play a role in the gene regulation. And also an important concept that was the base of that lecture is that cell differentiation results from gene expression regulation and not from gene deletion, which looks like obvious but is not. And some of you here, I see, have a paper, the seminal paper demonstrating that in 1962 as an exercise for Journal Club. So if you want to go to that paper, just ask. So, yeah, so have we some people that are feeling super no, but none are bad, so that's perfect. So let's try to wake you up and get the so-so on the way to the super, hopefully. Okay, that's a big task. So today, we will see the next step about regulation of gene expression that makes a cell specialized. And particularly, we will look at gene accessibility. And then we will also look at cell memory. Okay, so there are, in the eukaryotic cells, in the eukaryotic environment, you have most of the genes that at the steady state are repressed. So we are all under control towards a repression of gene expression. And this is achieved mainly by a lack of accessibility of transcription factor and polymerase to DNA. And that's what we will cover today because of compacting of the DNA in the nucleosome and because of DNA methylation. And then once all that is open, the transcription factor can bind the specific one, the general one, and lead to gene expression. So about chromatin structure, I think you certainly have already seen that somehow. And what's the purpose of those lectures? To integrate all this in the context of cell differentiation. But yeah, if it's too simple, just say, okay, you can move on. If it's not detailed enough, just also say, please stop. So about chromatin structure, I guess you know that the DNA is packed and is around histone making nucleosome, and you have octamer of histone that makes this unit of nucleosome. This has 2 main purposes. First is to get 2 meters packed in a few micrometers. You need to pack seriously that DNA to fit it into a few micrometers. And second, it's a major mechanism to regulate gene expression. So this is a microscopic picture, and you can see what is this. Initially, it was described as euchromatin and was described as heterochromatin. Well, people were seeing that DNA was in different form structure, but they didn't know what it corresponded to. And then later on, it was found that this form of open chromatin versus compact chromatin had different outcomes for gene expression. So how is this all controlled? The packing of DNA, am I too quick, or is that okay? It's okay? Yeah? Still awake? So the DNA is packed around histone, and this can be modulated by two main ways. These two main ways are through histone modifier proteins and through chromatin remodeling complex. So we will go through both of those mechanisms that are summarized on these slides, but we will go through that now. So there is possibility to acetylate histone, to methylate it. These are the main mechanisms that are studied and described, but the histone can be also phosphorylated and also be ubiquitinated, which, as you can know, leads to degradation. And then there is this famous chromatin remodeling complex that can move the histone along the DNA, but we see that now. So as you all know, a DNA is heavily negatively charged, and the histone are positively charged. So once they are bound together, there is tight and strong interaction, and the DNA is so packed that transcription factor cannot reach to that DNA and bind and induce all the sequence reaching to the formation of the big complex and polymerase activation. So once you have a histone modifier protein that is bound, you can have acetylation, for instance, and this will make the histone negative and then the chromatin structure open. This is a direct effect of those histone modifier protein. Then you have an indirect effect, because once the histone have been, for instance, modified, have become acetylated, then there are other proteins which didn't have affinity for the histone themselves, but have affinity for the histone once they are acetylated. So these are called the histone modification protein. I don't know why, I don't find that's a perfect name, but that's the name it is. So there are proteins called histone modification protein. They can bind only to methylated histone or only to acetylated histone. And once they are bound, this one can recruit other modifier, another acetylase, another kinase, whatever, that will affect the structure. All these proteins that are bound to the modified histone can also recruit this famous remodeling complex. Is that okay? Yeah. So I'm sorry I don't find that image very nice, I would say, but I could not find a better image of this chromatin remodeling complex. Also look for video, but yeah, nothing that clear. Well, the principle is that you have a complex, and this requires energy, that will bind to the DNA, and we will see how, and that will just move the nucleosome and make the site for binding of transcription factor accessible. So you see here we are in a resting state. The site for binding for transcription factor for the specific one or for the general one are not accessible. And then you have this remodeling complex that can bind and will move the nucleosome and make the site accessible. This can happen to sliding, as is shown here, but they can just expel the nucleosome or make it go to another DNA strand. And this requires energy. How are you? Okay? Okay, so I think, yeah, that's the question from last year. I need to clear result. But you, okay, maybe you saw, but maybe you don't know. So the regulation of gene expression, you probably know, and we discussed that last time, that transcription factor act at the level of the DNA. They bind, they recruit coactivator, and then they allow RNA polymerase to work. And then the question was whether this can, okay, so that's changing mind here. Okay, that's interesting. So, okay, 2 and 2. Well, okay, I think you balance. So, yeah, so the main function that is described, or the one we insist on a lot, is that transcription factor will bind this enhancer sequence or repressor sequence on DNA, and then they will recruit all the molecule and lead to gene transcription. But we just see now that before doing that, you also need to have access to DNA. Well, in fact, the transcription factor, they contribute to that very first step as well. So the correct answer is C, because they not only lead to a regulation of the transcription, but also to the structure of the chromatin. I don't know why I need to move. Yeah, so you see my email. Maybe I will close this down. Okay. So how do the transcription factor do that? Sorry for the technical point. Well, okay. No. Okay, so they can also act by modulating the chromatin structure. This is summarized here, and I will show you that here. Again, a scheme that is not perfect. This is from the Albert textbook. So you have here the DNA that is packed around the histone, and then there is one part of the DNA where a transcription factor can bind. But at this stage, the chromatin is not open enough to recruit all the other coactivator. What will happen is that, in fact, this transcription factor can bind those famous histone modifier protein. So you have here a transcription factor that is bound to the enhancer sequence on the DNA that is barely accessible, and then this will allow the binding of a histone modifier protein like a histone acetylase. So this is showed here in yellow. This histone acetylase will acetylate histone, make negative, and open the structure. And then because you have some histone that are acetylate, you remember we have those famous histone modification protein that can now bind on an acetylated histone, for instance, and this will recruit another histone modifier. So it's a cascade. Once you start the process, because you have only few parts that are accessible, the transcription factor binds, recruit histone modifier protein, which in turn will recruit other histone modifier protein. It's okay? And also this famous chromatin remodeling complex, they cannot bind directly to DNA. So that was a step that I just missed in the previous slide to exert this famous, here the remodeling complex, they do not bind directly to DNA to move the nucleosome. In fact, they first need to bind to the transcription factor. They don't have affinity for DNA itself. They have affinity for another protein, such as the transcription factor. So those transcription factor, the first thing they can do is opening chromatin because of recruiting histone modifier protein or because recruiting the chromatin remodeling complex. And then they will go and bind to the sequence, recruit coactivator, and lead to a gene activation. You have a question on that? Or comment? Precision? It's okay. So we talk a lot about activation. Then here is a scheme summarizing the mechanism of repression. So the repression acts at the same level, the level of gene accessibility and the level of gene expression, so a transcription. So here you see all the effect that repressor can have on the formation of an initiation complex, allowing the RNA polymerase to work on. So they can just bind at the same site as an activator transcription factor. The repressor transcription factor can just compete to the binding site to the coactivator, or they can bind on the coactivator molecule and inhibit. But they can also recruit those famous chromatin remodeling complex and then let them move and make the activating sequence inaccessible. So it's exactly the same, but the opposite way. Okay? Yeah? I lost you here? It's okay. Okay, so that's a summary you could give me, maybe. Yeah, just summarizing what we saw first lecture, and now we can go through. That's very important information, is that every cell of a multicellular and a complex organism contain the same DNA sequence, and it's because of gene regulation that we find specialization. Yeah, last time I asked, but I didn't have a clear answer to that. So we discussed briefly how Mr. Gurdon proved 60 years ago that gene deletion is not responsible for specialization. That was because of transfer of nucleus of specialized cells, and that could restore the whole organism. Today, how would you show that there is no gene deletion? How would you show that? What was the technique you would use? Yeah, you could sequence the genome, and then you would see that every cell has the exact same DNA. So that's perfect. And also, I wanted to mention new technique that really address this specialization of cell, even in a tissue. So, for instance, you have a liver tissue. Not all the cells are the same. You have many, many different cells in the same tissue. So specialization is really extreme. The organ has a specialized function, but in one organ you have many, many different cells at different stages of activation. So the technique that were used, let's say, until 5 years ago, you were taking a tissue and you could look at which gene are expressed by doing transcriptomic analysis. You were looking at all the mRNA in that tissue. But this was a bit raw approach. You could not really address all the specialized cells that make your tissue unique. So now there are more and more techniques you may have heard, or you will hear about single cell mRNA sequencing. This way you will take your organ, split it in multiple cells, and analyze the gene expression of each cell of one tissue. And then you will see how complex is a tissue, and you could detect very slight abnormalities, detect one very rare cells, because you will really address the whole complexity. So that was a comment. So then we have the transcription factor. That first step will need to contribute to opening the chromatin, and they can do so because they can recruit the chromatin remodeling complex, or they can recruit a histone modifier protein that can acetylate, open it, and then the transcription factor will finally bind to the enhancer sequence if it's activation, recruit lots of molecules, bound the DNA, and allow interaction with RNA polymerase and activation. All good? Okay, another way to control the expression of gene is DNA methylation. Just need to drink. So DNA methylation is mostly occurring, as you probably know, on cytosine. Cytosine or cytosine or cytosine? How you pronounce that? Cytosine? Cytosine, thank you. Cytosine. That's why I always say that wrong. So now I know. So it's cytosine and nucleotide, not nucleotide. Okay, so the methylation occurs on the cytosine, and then that are next to guanosine sequence. So we say the CPG sequence. That's easier for me. How does this happen? And how this DNA methylation influence the gene expression and the gene transcription? Always the same sequence, either directly or indirectly. Directly, because the methylated DNA change the affinity of the transcription factor for the sequence, so the transcription factor cannot bind anymore to the enhancer sequence. That's a direct effect that you can see here. Methylated DNA inhibit the binding of transcription factor. Or indirectly, again, always the same, the methylated cytosine will have affinity for a protein called methylate cytosine, methylate CpG binding protein, and this methyl CpG binding protein will then recruit molecule that will inhibit opening of the nucleosome. So this the opposite of the acetylase is the deacetylase, make the histone again positively charged, and then the chromatin condense. So direct effect, indirect effect, recruitment of deacetylase, and then again a compacting of the DNA along the nucleosome. All good? Ah, c'est pas bien, hein? So close your eyes. But you probably all know that now. Yeah, so think about the answer, and then I should, have technical issue, apparently. I don't know what's happening. But that's a simple question, but I should have done that before. Voila. Okay, don't be influenced by... so you may remember that we saw those extreme cell specialized. So yeah, you saw two of you think, or four now think that all is correct. So these two very different cells, very specialized, one are different because they have different DNA sequence. No, that we know all the cells have the same DNA sequence. They have a different pattern of DNA methylation. That is probably quite possible. Different pattern of chromatin structure, set of transcription factor data expressed, and then, it's a BCD that were the correct. Thank you for the 6 that are playing the game. Now I have to go out and move like this. I don't know why it's blocked. This is after the lecture. Okay, so we know that the cell, now you know exactly why a cell has a specific function, because a set of protein is expressed, and we saw how flexible is this, although it's quite hard to study and to analyze. It's so important, this gene regulation, because it makes the whole adaptability of our body and flexibility, which is such an advantage compared to gene deletion. But okay, so all that work to get to a specialized cells, achieving a specialized function, you don't want to lose this specialization. Once the cell is dying, you would like to keep your liver, or your brain, a brain. Not that your brain becomes a skin cell, for instance. So how is this maintained? That's the whole about cell memory. And you see here some cell, and on Thursday I will be in Europe, so I won't be in 2 places. So Aquila will replace me and will show you how important is cell differentiation in the gut to replace, and how is this achieved through stem cell differentiation. But in every tissue, you have stem cells that can lead to replacement, and you have more advanced specialized cells that can divide and replace the dead one. So there are 2 options schematically, but that in fact happen at the same time. Once you have a very differentiated cell, it can still divide and generate cells that have the same function, or let's say the same phenotype, so they express the same protein. So therefore, there must be a memory here, how the daughter cells know what the mother was. Big question. And then also you can replace the dying cells by a constant source of new cell that will differentiate, and this is through the process of stem cells. That's what with Aquila will cover. So today, now we will go through memory. And I hope this one I did correctly. Yeah, so that's an open question, so you can just talk, or if you're too shy to talk, you can write your ideas about how do you think that a daughter cell receives information about specialization. How does this happen? And all the signals that were there initially to induce the differentiation have disappeared. You are in an environment where you are not at the stage of development, so how does this happen? You can also just talk if you want. Yeah? Yeah, so you will transfer the gene content, and you need to transfer the epigenetic, meaning all the pattern of gene expression. But how, have you an idea how all that is transferred? No? How would you say that all that is transferred? Well, I'm happy you don't know, because then maybe you're excited to know the next slides. Inheritance of the epigenome. Yeah, I think some have the slides and are just copying, so which is creative. Yeah, indeed, but how is that inherited? That's the whole point. How do you transfer that? We will see the trick of the body. So, yeah, so there is transfer of DNA, and then there is the pattern of gene expression. And that is, the name is epigenetic inheritance, or epigenetic cellular memory is the whole pattern of gene expression that is transferred to the daughter cell. How does this happen? Well, many people are looking at that because it's of course super important, and sometimes it's good, sometimes it's not good. Because imagine your grandmother has been exposed to a toxic, and then this has resulted in gene repression, but you live in an environment you don't have that toxic, so you don't want that gene repression anymore. So you would like to reopen. So you need to know what controls the transfer of a certain memory to adapt this. Can we work on this plasticity? And also how can we achieve it? For instance, you know that cancer cells in general are not differentiated. You would like to make them go again being differentiated, and you want to ensure the cell keep differentiated. So this is the kind of perspective of looking at that. So here is a few options to achieve this task. So imagine you have an undifferentiated cell, typically a stem cell. It's at the stage of certain development. Then there is a signal saying, giving the instruction to the cell to differentiate. Therefore, for instance, this will induce, because you have that growth factor, for instance, will induce the expression of a certain transcription factor, A. And then the good thing is that this transcription factor A is driving its own expression. So then it's easy, even though you would not have anymore the signal present in the environment, for instance, the growth factor, the differentiation factor is not there. The transcription factor that is driving its own synthesis is still there. So you just need, now you have here a specialized cell. This cell is dividing, and some of the transcription factor will go into the daughter cells and will lead to their own expression. So you just maintain the specialization by transfer of transcription factor, which exerts a positive feedback loop. It's okay? So that's quite a simple idea of the body. Then the inheritance of the pattern of DNA methylation. So we know that cytosine, I forgot, cytosine, this is really not cool, huh? Cytosine is methylated, C is methylated, and, but once you replicate the DNA, the new DNA strand is not methylated. So how do you get that strand methylated? Well, easy, just ask. There is a protein called the maintenance methyltransferase, And this has an affinity for DNA where cytosine is methylated. Then it will bind there and will just methylate the other cytosine that are in front. So that's a way to maintain this pattern of methylation. So now you have the two strands that are methylated and just where they were because they are just in front of the initial site of methylation. Good. So we have a feedback loop where some transcription factor induce a positive feedback and you don't need the initial differentiating event. You have a possibility to transfer the pattern of methylation of cytosine because of the specific affinity of the maintenance methyltransferase. Okay, and we also would like to have inheritance of the pattern of chromatin structure. How do we get that? Thanks to the methylation pattern. You all remember, I'm sure, this was 2 minutes ago, or 5. It was here. Yeah. You saw that when DNA is methylated, it can bind to some protein, the methyl CpG binding protein, and this one has an affinity for molecules that affect the chromatin opening, such as histone deacetylase. So you understand that if you succeed to transfer the pattern of DNA methylation, and that we just see it thanks to the maintenance methyltransferase. So we have already this, and now we know that because the methylation is on certain pattern, this will recruit the methyl CpG binding protein, this will recruit the acetylase, and this will open the chromatin on the same way. So you just start with the methylation pattern, and then you get the same deacetylation pattern. Yeah? Okay. Another possibility, and that's the last possibility, but then I will ask you to talk. So be ready. So now you also know that when there is DNA replication, the histone are transferred in the daughter cells, but of course you lack half of the histone. So only half of the histone will be transferred with the correct, meaning the one that you need for that cell, specific modification. So imagine here you have methylated histone. Then you transfer that methylated histone in the daughter cells. And then you have new histone that are not methylated. Okay, you would like that histone is also methylated because it was like this in the parental cell. So therefore, it's again the system of a positive feedback loop. You just need that having an histone that is, for instance, where is the arrow? The histone is methylated, will lead to the recruitment of this histone modification protein that will then recruit histone modifier protein that is also a methylator one. You see that? So once a histone is modified in a certain way, it will recruit protein that induce the same modification of the histone that are just next to it. So it's again a positive feedback loop. Okay, so let's go to that. Who would like to tell me the 3 main mechanisms by which cell memory is achieved? Oh. So you would like we go to something easier? Let's go to something easier. One I think you have all aware. Describe 2 mechanisms of DNA accessibility. What are the 2 main mechanisms that control DNA accessibility? Yeah? Yeah, so it's the chromatin structure. That's one thing. So the packing and unpacking of DNA around the nucleosome. And the other one is? methylation of DNA. Yeah, so that's perfect. One. How do transcription factor affect DNA accessibility? Do you remember that? But we can go through the slide, so I think it's good to go. So you remember how transcription factor, you know, they control gene transcription, but they also control DNA accessibility by two mechanisms. Yeah? Exactly. So those chromatin remodeling complex cannot bind directly to the DNA. They need to bind to a transcription factor, and they have the superpower to displace the histone. And also the transcription factor, they can bind an histone modifier protein, like an acetylase, that will open. So the transcription factor play a role in DNA accessibility two ways: the access, the methylation, the opening of the structure through the two different mechanisms. So yeah, I was already answering the three. No, that's done. Four. What is cell memory? Okay, what is cell memory? How would you define that? Typical stupid question of exam. What is cell memory now? How would you define that? Yeah? DNA modification, yeah, you mean, yeah. Okay, the DNA modification, so the DNA accessibility and the DNA methylation, yeah, perfect. Yeah, that's like, it's not, the memory is not the transfer of the DNA content, which is already transferred everywhere. The memory is about the gene pattern, the pattern of gene expression, and that is related to all what we discussed. So cell memory is transfer of pattern of gene expression through the transfer of, what did I say? I'm tired. The transfer of the pattern of gene expression through all the epigenetic modification that you have in a specialized cell. Okay. Yeah, I'm sure you're convinced that's very important, that if you don't have any cell memory, then you lose the function of your organ and you become a fetus one day. Maybe we would like. But yeah, some people try also to do that, to revert the clock and stay younger. And now the last one, how the three main mechanisms by which cell memory is achieved. We can go through if you want to. So that was the last slide we saw. Okay, so you can have this epigenetic inheritance because you transfer some transcription factor that exert a positive feedback loop. You can have the transfer of the DNA methylation pattern, and this is thanks to the maintenance methyltransferase protein. And then you remember, I'm sure, that once you have a certain pattern of DNA methylation, this will also affect the acetylation of the histone, because the methylated cytosine can recruit some molecules that will then lead to deacetylation. All is linked. And then you remember that another third way is that when a histone is modified in a certain way, it will recruit protein modifier that induce exactly the same modification of the histone. So you have, again, a feedback loop there. Yeah, so that's it. Happy to get questions. And so next Thursday, Aquila will present you talk to you about stem cell. It's at 8 a.m., but it's a super nice lecture, so try to come. And then you will have other lecture, and I will come back about we see the differentiation, and then we see the cell death, and we see the apoptosis and how it's work and how important it is. So thank you. And there will be Honors Project Expo, I think, on the 26th here in the school. So yeah, come and see the people and discuss and yeah, and ask questions when you want to by email or like this. Thank you. So, and when and add it, okay? And then you